Good morning. It looks like it's 8:30 Central Time to get rolling. I'm Carl Bradley. I'm an extension plant pathologist with the University of Kentucky. I'm based out of Princeton at the UKREC, and I'll be moderating today. A couple of things before we get started. I just want to explain the format. I know many of you may have been on the first day of the winter wheat meeting on Tuesday, so this is day 2, and so it'll be the same format as that. If you're joining us for the first time today, what we have is a group of speakers that will be presenting through recorded presentations, and then after that, we will have a panel of those speakers where we will all be here. If you have questions during the presentation, it's okay to go ahead and type those into the chat box. And if I see them, we'll try to get some of those addressed after their presentation is over, or you can wait until the panel and ask questions then as well. So we didn't have registration for this meeting, but we really want to know who has all joined us. So if you could, please, into the chat box, if you could please type your name, where you're from, location, city, state. That would be great so that we get an idea of who all is with us today. We will also, and I think already are, streaming live on the Kentucky Grains Facebook page. So if you know people that can't access Zoom but want to see this presentation, see the presentations live, you can let them know that it's on the Kentucky Grains Facebook page. The first day, the recordings from the first day are on the Kentucky Grains YouTube channel. Those have been uploaded now. So you can go to that website or just search for Kentucky Grains YouTube channel, and you should be able to find those presentations that are already uploaded. And then sometime after today, the day 2 presentations will also be on that channel as well. So just a quick idea of who we have today. Dean Cox, the Dean of the College of Ag, will be giving the introduction and welcome. Carrie Knott will be talking about current wheat crop conditions. Bill Bruning will be talking about wheat variety differences to Metribuzin tolerance. Dr. Raul Villanueva will be talking about insects in stored grain. I'll be talking about management of Fusarium head blight, and Travis Legleiter will be talking about annual ryegrass control in wheat. After the recorded presentations, we'll have Bill Bruning will be presenting the Wheat Science Service Award, and then we'll go directly into the panel after that. So, as far as breaks, you're on your own for that. Just be sure that you turn your mic off during the breaks, and actually be sure that your mic is off during the entire period, just to kind of help avoid any interruptions. So we'll go ahead and get started, and I will share the first video, which is welcome from Dean Cox.
Hello, I'm Nancy Cox, Vice President for Land Grant Engagement and Dean of the College of Agriculture, Food, and Environment at the University of Kentucky. As we all know so well, due to the ongoing pandemic, we have not been able to gather in person for many of our traditional signature events. However, amid this pandemic, as I look across our college, our university, and to our many friends and stakeholders, I'm reminded of the importance of partnership. That you are all joining us is indicative that our shared work is ongoing and ever more essential. As a land grant and flagship research university, a connection to the Commonwealth is deeply tied to our history. In my new title as the Vice President for Land Grant Engagement, I've spent time working across campus on new opportunities to partner. And at the center of those conversations is the deep and abiding engagement the College of Agriculture, Food, and Environment has with communities across Kentucky. Our partnerships with citizens of Kentucky helped attract a large milling company to locate and become a vital partner in growing wheat. Our university, Kentucky farmers, and the Kentucky wheat industry are committed to the sustainable and secure market that wheat provides to farm families and local businesses. We deeply appreciate the commitment of Seymour Milling Company for the $1 million towards the Grain and Forage Center of Excellence and the many projects relevant to wheat and the larger cropping system in our region. Our partnerships with the Kentucky Small Grain Growers, Seymour Milling Company, private crop scouts, county ag agents, and numerous farmers are essential to UK fulfilling its land grant mission. It is certainly my goal and a shared goal at UK to be the land grant university that does the most for its state, and these are gold standard examples of what is possible. I look forward to new opportunities that are on the horizon and creative ways we can expand engagement across Kentucky in the future. Thank you for joining us and enjoy the day.
All right, we surely appreciate Dean Cox and providing that welcome, and appreciate all of her efforts as well. So we're going to move on. Dr. Carrie Knott will be next. Dr. Carrie Knott is one of the extension agronomists. She's located at Princeton, and she's also the director of the UK REC, and she'll be talking about crop conditions.
This is Carrie Knott with the University of Kentucky. I'm the Grain Crops Extension Agronomist. I'm going to spend just a couple of moments discussing the current condition of our winter wheat crop here in Kentucky. Thankfully, what I'm hearing from agents and producers, and also what I'm seeing across the state, is some very good winter wheat stands, similar to those in this photograph. And what you'll see here is that we have very good tillering per seed, a very lush stand, dark green. And so this is exactly an ideal stand of wheat going into winter dormancy. And so there are some producers that have reported some challenges getting winter wheat within the preferred planting window. And for those producers, whatever the delay may have been caused by, whether it be illness, equipment problems, resources, and also weather conditions, any of the wheat that got planted just a little late seems to be struggling just a bit. And this is more of a visual issue at this point. It's similar to what I'm showing here in that it's sort of thin stands. There's not a whole lot of tillers. You can see that there are a decent amount of stems, but far cry from the lush sort of stands that we would like going into winter dormancy. However, for those producers that had to plant late, either all or a portion of their crop, don't lose hope. As always, winter wheat is a very resilient crop. It's hard at this stage to really count it out. For those wheat fields that did enter winter dormancy with a little bit of a thin stand like this one shown here, don't despair. We do have options. Prior to Feeks 3, just prior to Feeks 3 or green up in the spring, what you'll want to do is go out and count the number of plants per square foot. If you have less than 70, you probably want to consider a split application of nitrogen. And for the first application at Feeks 3, you will likely want to apply half of your nitrogen. The remaining half of your nitrogen, you're going to want to apply around Feeks 5. This differs from a normal split application where you put on a third at Feeks 3 and 2/3 at Feeks 5. However, this elevated application of 50% at Feeks 3 will help with promoting some tillering early in the spring, and therefore possibly help mitigate any yield loss due to these thin stands going into winter dormancy. Other than that, I'm very optimistic for this year's season. I'm hopeful that we will have a bin busting year, and I do very much look forward to our winter wheat production year and getting back out in the field. Thank you very much. This has been Carrie Knott with the University of Kentucky.
All right. Thank you for that, Carrie. And again, if you have questions, you're free to go ahead and put them in the chat box, but we will have a live panel after all the presentations. So that would be another opportunity for you to ask questions as well. Next, we have Bill Bruning, who will be talking about wheat variety differences to Metribuzin tolerance. As you probably know, Bill coordinates the UK small grain variety testing program.
Well, good morning. I'm Bill Bruning, UK Research Specialist and Coordinator of the Small Grain Variety Testing Program. Today I'm going to be talking about wheat varietal differences in Metribuzin tolerance. And wheat varieties have been shown to be potentially susceptible to Metribuzin herbicide. And Metribuzin has been shown to be an effective control of ALS-resistant weeds, particularly or most notably chickweed. But it's also an effective control for annual bluegrass and Italian ryegrass. It was released about 50 years ago under the Sencor brand name by Bayer. So it's most commonly associated with that brand name, but it's also has been released under the names Lexo and Tricor, Dimetric, among other names. And as I mentioned, wheat varieties do have a variable tolerance levels to this herbicide. And so tolerance ratings in general are useful for growers if they're considering Metribuzin as part of their management strategy, but certainly important to consultants and seed companies as well, who will be advising their growers. Ratings are important, certainly with current varieties, they were last evaluated in the United States in 2013 and 14 by Mississippi State University and the University of Tennessee. So we're well overdue for taking a look at how modern wheat varieties respond to Metribuzin herbicide. So I conducted 2 years of investigating this in a wheat variety trial, evaluating varieties for differences in Metribuzin tolerance. And in the 2018-19 season, we had 84 entries. Most of them were Republic varieties, but also some experimental lines that have potential to be released as varieties in the future. And in the 2020 harvest season, we had 88 entries. And the goal of the trial that I put together was to induce damage, to really see what effect does this herbicide have on the wheat varieties. So I applied the maximum labeled rate, which was 10 3/4 ounces of Dimetric EXT, which is a 75% Metribuzin compound. I applied that in late winter at approximately the Feeks 4 to 5 stage. In the first year, that was, I believe, was in mid-March, and in 2020, it happened to be, the stage was in mid-February. And I sprayed half of the plot, and I did this so I could do a side-by-side visual evaluation to do a damage ratings, visual damage ratings. And I took these injury ratings at 3 to 4 weeks after the herbicide application. And also throughout the growing season, I recorded plant height and the heading date. And then at maturity, we harvested the plots, looked at grain yield and test weight. So for the 2019 results, you can see we had our 84 entries here. And I had an injury scale from 1 to 5, where 1, there was absolutely no sign of injury or stunting or discoloration at all. And the 5 rating showed severe injury, plant death, severe necrosis. And this plot here is actually a picture of the plot that had the 5 rating. There was only one variety that had the severe rating, and you could see there was actual just plant death compared to as we get into the more healthier non-sprayed area. There were a few other varieties that showed severe damage. I didn't see plant death with the 4.0 or 4.5 ratings, but certainly some leaf necrosis and severe stunting at this level. But then if we see most of the varieties in terms of the frequency of distribution fell within the 2.0 to 3.5 injury rating. So what we're seeing here is a broad scale of varietal differences in the response to the Metribuzin herbicide. In terms of what effect it had on yield, we can see that for higher injury levels, we had much greater reductions in yield. Now remember, only half the plot was sprayed, so these reductions are potentially twice as much as what we're seeing. So we're really talking in this deal here, really probably 80% yield reduction, maybe if not more. And as you can see, varieties that are more tolerant to it showed actually a yield increase here. And I don't believe this yield increase was due to the Metribuzin herbicide itself, but actually a function of the fact that in this particular year we had this set up in a split plot design. So certain blocks were sprayed with herbicide and the control blocks were not sprayed. Well, it just have happened that one of the control blocks was in a wetter area of the field. And I believe it was that wetness that actually reduced the control yield as opposed to the treatment actually boosting the yields here. So I think the important thing to see here is really that at the higher injury levels, you did see yield reductions. In terms of grain test weight, there was really no difference based on the injury ratings. We did see height reductions associated with higher levels of injury. Very little or no effect at the lower levels of injury, but dramatic reductions in plant height. And we saw that there were differences in heading date, which is an estimate of plant maturity. And we saw that that maturity was actually delayed with the higher levels of injury rating. So if we look at the next year's data, 2020 results, and we had 88 different entries. And then this year, I still have the 1 to 5 scale, but I did not have any plots that fell within the severe 5 rating. The highest I got was a 4.5 rating, and that was just one entry. And that entry was actually the same variety that we saw here with the 5.0 rating as well. So in 2020, we just did not have as severe damage. I'm not sure if that was a function of the environment that was growing in, the weather, the climate, or something to do with the timing of the actual application itself. But you can see among the distribution of the ratings, most of the ratings fell within the 1 to 2.0 rating, which is no to essentially just minimal stunting here. So not much damage, but we did see a number of varieties, and they were most of these did coincide with these varieties over here that we saw, the more severely damaged ones in 19. So it was repeatable from year to year, that response. But the yield, we saw yield reductions as we saw in the year before, although not nearly as severe. Obviously, we're talking 30 to 40% here. We're only talking 5 to 10% here. So much as we saw less visual injury, we also represented itself with smaller differences in yield or yield reductions. Again, there was no effect on grain test weight associated with the herbicide, but we did again see that there was stunting associated with the herbicide application. The higher injury levels showed a higher level of stunting, lower plant height here. And again, we saw that heading date or plant maturity was delayed at the higher levels of injury. So the take home from this, the conclusions of this 2-year study is that there's a wide range in tolerance among varieties, and that this herbicide can reduce yield, plant height, and delay maturity. And when considering using Metribuzin, it's important to check varietal tolerance. And this data is available in the UK Small Grain Variety Test Report. And with that, I'll be happy to answer any questions.
All right, appreciate that, Bill. Again, if you have any questions, be sure to type them into the chat box, and we'll have that panel after all the speakers. And if you joined us late, we were just asking everybody to go ahead and put their name and where they're from in the chat box as well, so that we know who all has been able to join us today. Next is Dr. Raul Villanueva, who'll be talking about management of insects in stored grain.
Hi, my name is Raul Villanueva. I am the extension entomologist at Princeton. And Dr. Sam McNeil also provides some collaboration with this presentation. The topics include insects in stored wheat, the most important, and considerations to reduce their populations. These considerations include prevention and also what kind of treatments we can conduct. Beetles are one of the most important pests for stored grain. So we have a couple of them. Here I'm showing you the red flower beetle and the confused flower beetle. These are very similar species, and the name of the second is due to the confusion on the species at the beginning. So you can differentiate these two insects based on the antennal clubs, the end of the antennal clubs. So the red flower beetle has 3 clubs that are the same size, whereas the confused beetle has 4 clubs and they are increasing in size. Also, the area that is between the head and the wings, these are called the prothorax. This area on the red flower beetle is kind of curvilinear. You can see here, whereas in the confused beetle it's kind of straight or direct line. Both of these species can be very important, especially on processed food. However, the red flower beetle is a serious pest in storage bins in farms because they lay sticky eggs on sacks, in cracks, or walls, and they are sometimes hard to dislodge. On the other hand, the confused flower beetle is a serious pest in flour mills and in warehouses. So both of these insects can be very easily found in pantries, especially when you left food storage for a long time. There is another beetle that's very important. This is the lesser grain borer, and you see that it has a cylindrical body shape, and the head is beneath the prothorax. This insect can lay single females can lay up to 300 eggs around the grains or in the grains. And this is a primary feeder. This means that they can damage healthy grains. Both adults and larvae feed on the grain, and the mandibles of this insect can perforate wood or paper. So they are sometimes hard to distinguish when grains are damaged. Another group of beetles are the weevils or snout beetles. And you can see here, this beetle has a very elongated prolongation on the head. And at the end of this prolongation, you can see the mouth parts that can use to come from the grains or to perforate grains. So we have several species. We are showing here two, the Granary weevil and the Maize weevil. These two species do not fly, but there are some differences among them. The Granary weevil is kind of reddish in color, whereas the Maize weevil is kind of dark, and it's a little bit larger. In addition to that, if you see here the prothorax on the Granary weevil, the pits are kind of elongated, elliptical, whereas in the Maize weevil, they are kind of circular. On the other hand, the rice weevil is probably one of the most important species because it flies, so when it flies, it can disperse very easily, whereas the other ones are transported within the grain. So there are some differences between between rice weevil and the Maize weevil. They are kind of similar. However, the rice weevil has very distinctive yellow marks in the elytra here. But in addition, also they have a similar kind of pits, circular ones. Another distinctive characteristic is here the middle line between the elytra. So they are not contiguous compared to the Maize weevil. That's kind of the ridge is there, but it's not very visible. Rice weevils are very destructive insects in storing grain. As you're going to see, they are probably one of the most abundant in Kentucky. So the larva lives inside the grain, so they complete the entire life cycle inside. And if the grains are stored for a long time, they can produce outbreaks. So the temperatures are very important. For example, at 86 Fahrenheit, they can complete the life cycle in 30 days. At 77 degrees Fahrenheit, they increase 10 days more. So this means that a pair of weevils may produce up to 6,000 offsprings per year. The adults are short-lived. They don't feed. They just emerge, mate, and start to lay eggs. On corn, these rice weevils can develop 3 specimens within one kernel, whereas in wheat there's only 1. Moths or lepidopterans are also important pests in small grains. One of the most important is the Indian meal moth, Cytotroga cerealela. This is a moth, very tiny, but very colorful. As you can see, they have some bands that are kind of reddish and black, very easy to be recognized. They have a very rapid life cycle. They complete they have a development time on fewer than 4 weeks. In addition to that, it has many hosts. They have dried nuts, grain, many processed food, and of course, grains. The larvae of this insect produce a silk that, while it's growing, it's producing great numbers, but also it protects them from pesticides. So this, when you are trying to move the grain, you notice this silk, and this reduces the prices of the grains. In addition, the excretion from these moths also causes contamination on the grains. So the insect cannot survive to temperatures below 50 degrees, but above these temperatures they can start to feed and reproduce. Also, we have the angumized grain moth, although this is more commonly found on grains. Before moving into insecticides, we need to consider some preventive methods to reduce populations or infestations of insects on stored grains. So one of them, very important, is have safety situations. So you need to use the proper personal protective equipment. In the internet, you can find many videos where you see these kind of situations. This is not doable. So the worker here, he needs to use a proper protective equipment, harnesses and so on. Here you see Dr. Sam McNeil using this proper equipment when we did a visit in 2020. So he uses masks, head protection, gloves, and proper harness. So preventing also includes checking inside and outside structures, ducts, and doors. The proper sanitation before putting new grain is extremely important. We need to have beans completely clean. Avoid spilled grains inside and outside. Also, if you have a very small amount of grain, this can be a source of infestation. But not only that, you need to clean the tracks, you need to clean the big augers, and even before doing harvesting, you need to do a very good cleaning of the combine. Otherwise, you are bringing infested seeds to your beans. Also, you need to consider finding sources of infestation. You need to try to check up for every small holes within the structure. And if you don't repair them, for example, in this case, if you don't repair this one, so you are not going to be able to find, you are not going to be able to control the insects and the rodents also. So in addition, you need to do a proper identification of the insect, because otherwise you are going to use insecticide that is not going to control the pest. Although controlling environmental conditions is very hard, especially during the summer when we have wheat, the periodical run of the dryers are important. And especially this is the case during the winter when we have some sudden temperatures above 60 degrees. If you run the fans, they can stop weevils or the Indian meal moth to fly. So in the winter especially, we need to have temperatures below 50 degrees. In the summer, that's a little hard. We need to go move. We need to do more spray with insecticides. So in 2020, we are starting to monitor insects thanks to funds provided by the Siemens Miller company, and we have collected a couple of them. And in addition, we have right now a colony of weevils and moths. So with that, we are going to do some tests. But in addition, also in 2007, we have some sampling, and we found there in 2017, the weevils were present in some beans, but in others, for example, in the second bean, you see that 56% were weevils. And in 2020, we started to do the same. On November, 68% of the insects captured were weevils, and on December, 67%. So that's the reason we are going to start doing the monitoring with, we are going to do the insecticide tests for weevils. So all the information that I'm going to present in the next slides, you can find on the Entomology 47 Small Grains PDF that is freely accessed in the internet. There are several options when you do a cleaning of stored areas. The first one is clean out with fumigants, and these are done when the beans are empty. So in this case, you apply based on the volume that this bin contains. You can use aluminum phosphide tablets or pellets. The rates are there, and these are expressed on per 1,000 cubic feet. And you can apply this product to boots of elevators, beneath false doors, and so on. On empty bin surfaces, you can do also a surface application. And this is calculated based on the area of the bin. You have several products: Sentinel, Diacon, Pyronil, Storicide, Insecto, and Tempo. Remember, Tempo should be applied only to the structure, not to the grain. In addition, you need to wear a protective equipment, especially when you use Diacon or Insecto. These are dust-based formulations that can be toxic. If you have bins filled with grains, you can use several products such as Sentinel, Diacon, Pyronil, Sensat, Storicide, and Insecto. So remember, these are products that you can apply directly to the small grains. And the amounts, the rates are based on the weight of the grains. Remember also that there are different products for wheat, different rates for wheat or for barley. And in addition, you should try to avoid using the same compound for bin surface or for grain protection. It's better to change the compounds. For grain surface or cap out, you can use Biopit, Dipel, Diacon, Pyronil, Sensat, and also Vapona. So these products are calculated based on the area of the to be treated. Remember, Biopid and Dipel, these are Bacillus thuringiensis base, and they are specific for Indian meal moth, and they are not going to be effective for beetles. Also, Vapona, this is calculated for 1,000 cubic feet of air per strip, and this treatment lasts 3 months. For bulk grain protection, you can use fumigants. Fumigants here are applied to the stored grain, and this is calculated based on the weight of the grain that you have in a bin. So you can use aluminum phosphide as well as the same products that you use for clean out. However, because you are using on grain, the rates here are a little bit higher for tablets and for pellets. Compare this: tablets, for example, 40 to 180 for bulk grain, whereas for clean out, you have 30 to 140. So I will conclude my presentation. Again, thanks to the Zimmer Milling grant that we are conducting these studies, and Caleb Whitney that is conducting the monitoring of insects. Thank you.
Okay, thank you, Raul. I want to mention that you saw Sam McNeil's name at the beginning of the presentation, and then there was even a really good picture of Sam all dressed up in PPE. And actually, that's what Sam's been looking like lately coming into work. He was well prepared for the pandemic since he had all that equipment. So anyway, Sam is also on the meeting today, and he will be joining us as part of the panel. So if you have questions for Sam, who's an extension ag engineer, feel free to ask those questions as well. One thing I want to note is I cannot see the chat box the way that my monitor configuration is set up while I share my screen. So I understand that there is a question that's in there, but I think unless I think it was addressed to Bill Bruning, unless he's answering it via the chat box. I think we'll just wait to address that for the panel. So I apologize for that. The next speaker is yours truly. So if you feel the urge to take a break, we kind of set this up now so that it would be a good time to take a break while I'm speaking, because I'm, obviously we have a lot of good speakers here, and I'm probably the last one on the list you want to hear from. But anyway, I'm next, so we'll go ahead and start that video. Hi, I'm Carl Bradley. I'm an extension plant pathologist with the University of Kentucky. I'm based here in Princeton and cover diseases of small grains and soybean. Today I'm going to talk about Fusarium head blight, or sometimes we call it scab, of wheat, and how to manage that using integrated methods. One of the things that there's been more questions about recently is when is the best timing to apply a fungicide for head scab. And so for a number of years in a lot of our trials that we've conducted here at the University of Kentucky, as well as nationally across several different states, have really been focused on Feeks 1051, as you can see in the picture here, which is when anthesis starts, when those anthers start to emerge out of the middle part of the head. And we've seen in our trials, as well as national trials, that we often can get a similar effect of spraying the same fungicide up to 5 days later after Feeks 1051. So that window is really, for most of our data we've seen, is really starts at Feeks 1051 up through about 5 days later. More recently, there's been some talk about earlier applications. This has really come along with the new fungicide Metribuzin Ace from Syngenta, where one of the marketing timings for that fungicide has been Feeks 10-3, which is when half of the head emerges. I want to show you some research results from some field trials that we've conducted here at the University of Kentucky, as well as show you some data from some trials that have been conducted nationally across multiple states. In this data set, these trials are conducted under mist irrigation to promote scab, promote infection, and the level of disease that we have out there. The national trials are funded by the U.S. Wheat and Barley Scab Initiative. What I'm going to show you was conducted from 2018 to 2020. And then I'm also going to show you some trials that are just from the University of Kentucky that were funded by the Kentucky Small Grain Growers Association, conducted from 2019 to 2020. I should also mention that the national trials that I'll show you includes data from Kentucky as well as from several states. So the first results that I'll show you here are from those national trials. Again, these are from 2018 to 2020, and the data that you see here represent 47 different environments. So there's a number of different states that are involved here. Some states may have more than one location. And again, these are all mist irrigated and conducted in a way so that we have a lot of scab out here to evaluate. So these trials were supported by the U.S. Wheat and Barley Scab Initiative, and Dr. Pierce Paul at the Ohio State University is the one that sort of collects all the data and has summarized these results. So what you're looking at here is Fusarium head blight index. So this would really relate to the symptoms that you're seeing out in the field, the number of blighted heads and how severe those symptoms are on each head. So the bigger the bar, the more disease there was, basically. So this black bar here is our non-treated check. Then we had Metribuzin Ace applied at Feeks 10-3. And then the treatments here in the gray bars are all applied at Feeks 10-5-1, right at early anthesis. So here's Metribuzin Ace, Fusarium, and Carumba. And the letters on top of each of these bars show you the significant or statistically significant differences among treatments. So any letter, any bar that shares the same letter are not considered to be significantly different. So you can see the non-treated check up here. All of the other treatments have a different letter, so they all effectively reduced Fusarium head blight. Metribuzin Ace at Feeks 10-3 was basically the same as applying Fusarium or Carumba,
at Feeks 10.5.1. When Metribuzin was applied at Feeks 10.5.1, we saw an additional reduction in Fusarium head blight. So in this slide, we're looking at DON or deoxynivalenol or vomitoxin in parts per million here. As you may know, DON is regulated by the FDA, and when you're going to take your grain to an elevator, basically anytime you're at 2 parts per million or above, you're likely to be docked or sometimes even outright rejected, depending on how high the DON levels are. So we want to try to get below this 2 part per million level. So again, this is over 47 environments across the last 3 years. You can see the non-treated check was up here at 5 parts per million. One thing that's a little different from the last slide is this Metribuzin, when applied at 10.3, although there was a significant reduction in DON, it wasn't nearly as big as if you would have applied Metribuzin at 10.5.1 a little later, which would be the same timing where we typically see Prosaro or Carumba being applied. So you can see that Prosaro and Carumba and Metribuzin all significantly reduced DON levels compared to both Metribuzin applied at 10.3 as well as our non-treated check. Now I want to focus on some trials that were funded by the Kentucky Small Grain Growers Association. So these are trials that are just from Princeton, Kentucky here. These were also under mist irrigation. And these trials are from the last 2 years, 2019 and 2020. And what we're looking at here is Fusarium head blight index. So again, this would be the symptoms, and the larger the bar, the more disease there was. But we have a number of different treatments that we're looking at. This black bar is our untreated check. So you can see we had a pretty good level of disease there. And as you look across all these bars, you see no matter what we did, at least we had reduced disease whenever we applied something, no matter when it was or what it was. All these blue bars here represent products that were applied at Feeks 10.3 when half the head was emerged. And then the gray bars would be at Feeks 10.5.1, early anthesis. And then the green bars would be applied 5 days after we made the 10.5.1 application. So that 5DAA stands for 5 days after anthesis. Now, even though Metribuzin is really the only product that's being marketed to apply at Feeks 10.3, we went ahead and evaluated Prosaro, Carumba, and Folicure at that same timing. And you can see that Metribuzin has a little bit better reduction in Fusarium head blight whenever you're making that application. But none of the other products are really marketed to apply that early. So when we look at the 10.5.1 over here, these gray bars, we can see that everything shares the letter D. So there are no significant differences when we look across those different products that were all applied at that Feeks 10.5.1 timing. And then the green bar would have been 5 days later, and they all share the same set of letters as well. So no differences within this timing. One thing that you can just kind of see a trend, and although it sort of changes product by product, but in general, Feeks 10.3 seems to be a little bit on the early side. You're getting a little more consistent control with all of these products whenever you're applying at Feeks 10.5.1 or maybe just a few days later. Now we're looking at the effect of the different fungicides and different application timings on DON, on the toxin that contaminates the grain. And you can see we had pretty high levels of DON in this trial. Our untreated check over the last couple of years was over 6 parts per million, so that's pretty high. Again, that 2 part per million is sort of that threshold we want to try to be under. And as you look across there, none of these met that. So again, we're using a susceptible variety, and the only thing we're really doing here to manage scab is just applying a fungicide. So that shows you that sometimes is not enough on its own when you have an environment that's really favorable for scab. So here's the blue bars are that 10.3 application timing, and you can see that we did in some cases get a reduction, significant reduction in DON relative to our non-treated check. But our biggest reductions in DON occurred whenever we were applying at either Feeks 10.5.1 or 5 days after that. And you can see as you look across the gray bars and the blue bars, is that they're pretty similar, kind of have the same trend across both of those different application timings. And you can see that they all share the letter D, so they're not significantly different from each other, but they are significantly different from some of the treatments over here that were applied at Feeks 10.3, and definitely significantly different than our non-treated check. So what did we find out from these fungicide trials that were conducted both nationally as well as here at the University of Kentucky? I think for the Feeks 10.3 application timing, again, just as a reminder, Metribuzin is sometimes marketed to apply at that timing. It did provide control of symptoms of what we call the Fusarium head blight index, similar to other products that were applied at Feeks 10.5.1. However, when you're looking at dawn control, it looks like sometimes that may be a little bit too early, and that product is a lot more effective when you're making an application at Feeks 10.5.1. And just across the board with all products, we really found that the most effective treatments were those that consistently reduced Fusarium head blight and dawn were those that were applied at Feeks 10.5.1 or slightly later, up to 5 days after anthesis. In our research trials, we found that it was very similar to applying at Feeks right at Feeks 10.5.1. So that window of application is a little bit larger, but that window looks like it really starts at Feeks 10.5.1 if you want to get the best level of control. So, the title of my presentation has integrated management within it. And so that's one of the things I really want to focus on. And as you could see from our previous work, just looking at fungicides, sometimes fungicides just wasn't enough especially when we're looking at dawn levels in grain. So I want to show you some fungicide by variety trials where we look at both combinations of fungicide and varieties. These trials were funded by the U.S. Wheat and Barley Scab Initiative. Although we've been doing these type of trials for a number of years, I'm just going to show you data from 2019 and 2020, since that set of data includes Metribuzin, and these were all conducted at Princeton. In these trials, we do not irrigate. We rely on natural rainfall. We do, however, plant in no-till into corn stubble. So there is some level of disease out there. We also, in some cases, we may spray spores of the Fusarium head blight fungus on the wheat heads as well, just to try to increase our level of disease. In these trials, we had 3 varieties that ranged in their susceptibility to Fusarium head blight, and then we looked at some different fungicide treatments. We had a non-treated check, Metribuzin at Feeks 10.3, Metribuzin at Feeks 10.5.1, and then Prosaro at Feeks 10.5.1. So in this slide, we're looking at Fusarium head blight index. Again, the bigger the bar, the more disease, the more symptoms there were out there in the field. These trials were funded by the U.S. Wheat and Barley Scab Initiative, and they were from Princeton, Kentucky in 2019 to 2020. We have our 3 different varieties down here at the bottom. Agramax 446 was our most susceptible variety in this trial. Then we had Pembroke 16, which was sort of in the middle. And then Agramax 463, which was our most resistant variety that we had in this trial. The blue bar was our non-treated check. The orange bar was Metribuzin at Feeks 10.3. Gray bar was Metribuzin at 10.5.1. And then the yellow bar was Prosaro at Feeks 10.5.1. And you can see here with our most susceptible variety, it didn't matter which fungicide or which timing we used. We had a reduction in scab relative to our untreated check. And you could say that also for our variety that was sort of in the middle, moderately susceptible. And then our moderately resistant variety is Agramax 463. We see all C's across all these treatments because you can see that we started off at a pretty low level of scab, even in our untreated check. So this just shows a lot. This slide shows a lot for just using a resistant variety. You can see how just looking at the untreated checks here from this variety to the next, we had a pretty big reduction and a really big reduction when we used the most resistant variety. Here we're looking at the effect of the fungicides and varieties on dawn levels. Again, just looking at across the varieties, our most susceptible was almost, about a 2.8 or so parts per million. And then our moderately susceptible was 1.5. and then our most resistant was down to 1. So again, just using a resistant variety goes a pretty long way here. So looking at the different treatments within our susceptible variety, similar to what we had seen in some of our other trials, that Metribuzin applied at 10.3. You see some numerical reduction here, but not a statistically significant reduction in DAWN when we apply that early. However, when you're applying at Metribuzin at Feeks 10.5.1, we did see that reduction, a better reduction in DAWN, which was similar to what we saw with Fusarium applied at 10.5.1. Kind of a similar trend here with the Pembroke 2016, although in this case, all 3 treatments, fungicide treatments, had a similar reduction in DAWN. And then no differences across fungicides when we look at this most resistant variety, just because DAWN levels are already pretty low, and so we're not detecting any significant differences across those different treatments. So as we can see from some of the results that I've shown today, fungicide application by itself often is not always enough to achieve adequate control of Fusarium head blight and DAWN just on its own. So it's important that we use integrated management of Fusarium head blight and DAWN. The most important out of all these steps is planting the most resistant variety that you can find. That is the most important, as we can see from basically most of the data that I showed today. And then applying an effective fungicide at that right timing. And what it looks like from most of the data is that right timing kind of starts at Feeks 10.5.1, and then you can kind of go from there up to maybe about 5 days later, depending on the fungicide labels. And then another step that's important, although I think not as important as the top 2 there, is planting wheat after non-host crop. So if you're planting wheat after soybean versus corn, then your risk of scab is going to go down a little bit. You're still going to have a risk, but it's still going to go down a little bit. But just keep in mind that most of our acres of wheat are probably planted after corn here in Kentucky and in even some of the states around us. And that when we're doing that risk of scab is always there, that inoculum is always there. So it's that much more important to start off with planting a good resistant variety and then applying an effective fungicide at that right timing. I just want to take a second to say thank you to the Kentucky Small Grain Growers Association and to the USDA ARS U.S. Wheat and Barley Scab Initiative, as well as multiple industry partners for financial support of my small grains pathology research program. Thank you.
Okay, thanks for hanging with us so far. Just a reminder, please be sure that you keep your microphone on mute just to avoid any interruptions. And so also, again, I apologize, I cannot see the chat box the way that I'm configured here with my monitors. So we'll get to anything in the chat box once we get to the panel. But be sure to go ahead and continue to be putting comments or questions into that. Our next speaker will be Dr. Travis Legleiter, who's our extension weed scientist here in Princeton, and he'll be talking about annual ryegrass control in wheat.
Hey guys, it's Travis Legleiter, Extension Weed Scientist at the University of Kentucky. And what I want to talk to you guys about today, no surprise, I want to talk to you guys about ryegrass in wheat and how do we best manage that? How do we best control that weed in wheat? So I wanted to kind of take advantage of the fact that we're doing this over video and just bring you right out into the field. The great thing about wheat is in the middle of the winter is a great time to be evaluating it for ryegrass control, especially when we're assessing our pre-emergence herbicide. So that's what I want to do today is just walk through a few plots looking at some of our pre-emergence options. And then what I want to do is also give you guys some highlights of some other non-chemical ryegrass control methods that we're looking at and that my graduate student is looking at. So first of all, let's go look at some herbicide treatments and some pre-emerge herbicide treatments. So here we are in the plots, and what we're going to look at here is some pre-emerge residual herbicides to help us in controlling annual ryegrass within our wheat. Before we start looking at these plots, I do want to review with you what those products are. And what we're looking at primarily is products that have the active ingredient pyroxasulfone. So those products are going to be Fierce, which can be applied 14 days pre-plant, Anthem Flex, which can be applied pre-emergence, delayed pre, or early post, and Zidua, which can be applied delayed pre or early post. Okay, so keeping those in mind and those timings, let's look just quickly here. First of all, I want to show you the untreated check. So here's the untreated check. You guys can see how much ryegrass is in there. I'll pan up so you can see the plots behind it and actually see that there is wheat in these plots. And you can see how heavy of an infestation of annual ryegrass we have here. So there's also some wild onion and some other, there's also a couple of broadleaf winter annuals out throughout here as well. But very, very heavy infestation of annual ryegrass. So let's go ahead and look at that first treatment. Here's the first plot. And what this plot is Fierce applied 14 days pre-plant. Okay, so let's go ahead and take a look at this. And what you guys are going to see here is there are ryegrass escapes throughout here. So remember, this is Fierce 14 days pre-plant. So the Fierce was applied in early October, and then this wheat was planted on October 22nd. So keep those in mind, okay? And now what we're going to do, and so that looks a lot better than that untreated check, but for ryegrass probably still isn't acceptable and is still going to need something over the top or post-emergence to help control that ryegrass, okay? So we do have some escapes there. Now, right next door, what you have here, and as we walk in here, you guys can see this is a much cleaner plot. If you look in there really close, we can find a few ryegrass escapes in this one, but not as nearly as much as that last plot. And what this one has in it, or what this received, was Anthem Flex pre-emergence, okay? So between these two treatments, and this treatment also received finesse cereal and fallow, but we don't consider that as really helping us out with the ryegrass. But when we compare the two of these, we have on your left, we have the Fierce, and on the right, we have your Anthem Flex. Both of these products rely on pyroxasulfone as the residual to hold back the ryegrass. So these are equal in that right. Where they differ was the timing. The Fierce is allowed to be put on 14 days pre-plant, whereas the Anthem Flex was allowed to be put on pre-emergence. Okay? If you guys remember back to October, early October, we did not have any rain. It was dry, right? When that Fierce went on, we did not get an activating rainfall. A lot of this ryegrass began to emerge, and without an activating rainfall, that herbicide was not doing a whole lot for us. Okay. And then right before we planted, we got some monsoons that came through here. We had some pretty wet soils. We actually got delayed a little bit on planting this week, but when we got it planted, it was nice wet soils, not wet, but nice moist soils. And then we got a nice activating rainfall right after the Anthem Flex went on. And that's why it's working so much better. It was in perfect conditions, perfect scenario, a nice activating rainfall right after it went on. And that's why it's holding back the ryegrass. So again, like any residual, having activating rainfalls goes a long way. And that's where the Anthem Flex has a little bit of a leg up with the multiple timings that we can put it on. Let's go look at another option. One of those other timings that I mentioned for several of these products was early post-emergence in wheat. So 2 to 3 leaf wheat is what we're looking at. And this was Zidua that was applied to 2 to 3 leaf wheat, as well as, or included with that, 2 ounces of a dry Metribuzin. We include that Metribuzin in there because the Zidua will do nothing for any of the ryegrass that's emerged. And so that's what the Metribuzin is in there for, is to help control that ryegrass that's emerged, which certainly is going to be emerged and going to have a couple leaves on it if we've done nothing else in this field, which is the case here. And you guys can see, I've always been amazed how well that 2 ounces of Metribuzin can pick up quite a bit of ryegrass in that plot or in the field. So I know there are safety concerns with that, or concerns of crop injury. If we have 2 to 3 leaf wheat, we can usually avoid that. And as far as crop safety or injury risk with the pyroxasulfone, it's the lowest at this this stage. So with all these plots, this plot and all the plots that we just looked at, all of these do have escapes in them and have ryegrass that's going to need to be controlled with a post-emergence herbicide. And so usually we're going to do that with axial bold, and we've shown that works really well for us 95% of the time. But that control is going to start going down, right? We already know we have panoxidin resistance in the state, and we're pretty sure it's spreading fairly rapidly. So guys, we really have to start looking in the mirror and asking ourselves, what can we do besides chemical weed control for this weed and wheat? And so that's where it leads us next, is thinking about what are some of those alternatives. And one of those alternatives is harvest weed seed control. And so what I'm going to do is I'm going to turn this over to Amber Herman. She's going to talk to you guys about her project looking into this potential for ryegrass control in Kentucky wheat. Go ahead and take it away, Amber.
Hi, my name is Amber Herman. I'm a master's student here at the University of Kentucky, and I'm going to talk a little bit about my research where I look at harvest weed seed control options for Italian ryegrass in Kentucky. So harvest weed seed control is not something necessarily new, but it's new to the United States. It's popular over in Australia in their wheat production, where they have issues controlling rigid ryegrass, which is related to Italian ryegrass. It has similar issues, being resistant to many different herbicides, and it's very hard to control. They have roller mills attached to their harvesting equipment, which can grind up anything in the chaff, therefore destroying any seed in that chaff, which can reduce your weed seed bank. The weed seed retention in the shatter is what we're looking at, because this is vital to whether harvest weed seed control can be a success here in Kentucky. So looking at pre-harvest, prior to harvest of wheat, we looked at the seed brain. From the middle to late June, up to the point of wheat harvest, so roughly June 15th to June 25th, we were out looking at wheat fields and their level of infestation. And then for the 4 fields we selected that had heavy infestations, for every half acre that was infested with Italian ryegrass, we did a meter squared evaluation. In that evaluation, we collected the ryegrass seed heads in that square, and we counted them all. We also collected some wheat seed heads just to try and get a grain moisture analysis to try and see how close to harvest this field was. And then lastly, which is in this picture here, you can see that we collected any chaff, seed, and debris from the soil surface using a shot vac to try and see how much of the seed from the Italian ryegrass has already been shed to the ground. The results of these study, you can see I only have 2 locations there. It's because we only have 2 locations cleaned and analyzed. And we also analyzed these separately. But for both locations, you can see that there was more seed left on the seed heads than on the ground. For the Young Road site, there was approximately 11,000 seeds still on the seed heads, versus on the ground there was only roughly about 2,500. Now at the UKRAC site, we had roughly 1,000 seeds still on the seed heads again, but we had almost 4,000 seeds on the ground. And just to give you an idea of what these sites were like, the average seed head density, which means there was roughly 77 plants per meter squared at Young Road, and there was almost 125 averaged per meter squared at the UKRAC site. And as for these fields, then the Young Road averaged about a 23% moisture, and the UKRAC's wheat averaged about 16%. This is good going into the next part of my study. We were looking at harvest, then that seed that is still on the Italian ryegrass, will it shatter at the header, pass through the combine with the chaff, or contaminate the grain? We did this by using 4 1-acre blocks, and where we also did the pre-harvest seed study. There then, at the time of harvest, we laid out these small baking trays, almost the length of the header, as you can see here, to get an idea of when the combine passes over it, what is going to shatter there. So we had the combine come to full speed. Then once the header was over these trays, the combine stopped over them and then backed up. Anything that was in those trays then, any debris or hopefully Italian ryegrass seed, we collected so we could analyze how much seed was there. The second part was, again, the combine was allowed to come to full speed, and there was a person standing on each side of the combine. Once the header had passed them, they had 2 much larger trays to lay down, which was, again, a total of 4 trays, so that equaled an average of almost a meter squared area. They laid down their trays, and that collected any of the chaff that was coming out of the back. Then once the combine had completely passed them and there was no more chaff being deposited onto those trays, the combine stopped so that we could collect what was in those trays to sort through and analyze as well. Lastly, once the entire block was harvested and we did those samplings, we did a grain contamination check. We used a chambered grain probe to get a wheat sample from the tank. And from there, we could see how much Italian ryegrass seed contaminated into the grain tank as well. After doing all this, all those samples were clean, weighed, and then seeds within those samples were counted. This was done so we can kind of get an idea between pre-harvest, what shatters at the head, what's passing through the combine, and what's in the grain tank. We can give those percentages to see where majority of that seed is going to end up at harvest. So the results for this study show that roughly 7,000 seeds were in the chaff collection, roughly 5,000 were in the grain tank, and then we found in almost 4,500 in the head shedder. So this shows that most of the seed is entering the combine. The chaff collection and the grain tank were not significantly different, but that is where a majority of our seed is going. And then also the grain tank and the head shedder were not significantly different as well. But there, like I said, there is that difference between what is in the chaff and what is in the head shedder. And again, for this site, there was an average of 125 seed heads per meter squared, and this site was at an average moisture of 16%. To kind of wrap this all up, then you can see that a majority of the seeds are retained on the seed heads of Italian ryegrass up to the time of wheat harvest for this year, 2020, in Kentucky. Then from the at-harvest study we looked at, a majority of the seed was found either in the combine chaff or the grain tank during harvest. This leads us to believe that it is possible that a potential harvest weed seed control could be viable as an option for control of Italian ryegrass in Kentucky wheat. This will all be looked at again next year, and we hope to find out some more stuff from this. If you have any questions, I'll take them. Thank you. That's my email address, and I want to thank the Kentucky Small Grains Growers Association for the funding for this project.
All right. Thank you, Amber. And as you guys can see, there's a lot of potential here for Harvest Weed Seed Control to help us with annual ryegrass control in wheat. And we still have a lot of data to look at. We still have a lot of things to look into. But really, as an industry for wheat in Kentucky, we really need to seriously start looking at this as a potential option, as we know that we have panoxidant resistance, and we know we have a lot of fields that have ryegrass scapes at the end of the season. And really, we need to start implementing these types of technologies now, not when resistance is here, because as you guys can see, it's not that technology. Harvest Weed Seed Control doesn't give us 100% control. It's there to help supplement other programs. So we need to start implementing it now while some of our herbicides are still working, and that'll also extend the life of those herbicides as well. So we're going to keep looking into this technology and keep bringing you some information into it. And with that, Amber and I will be on here at the end to answer any questions you have. Also, here's my contact information if you have any questions after the meeting pertaining to weed control in wheat. Thanks for tuning in.
Well, thank you, Travis and Amber. That was very well done. Really thought that was cool how you could gives us an opportunity, I guess, doing it this way to really see what's going on out in the field. And it's pretty cool that you have some plots that you could show for this meeting. And both Travis and Amber will be on for the panel if you have questions for them. So we have one more recorded thing I want to show, and this will be from Bill Bruning, who will be presenting the Wheat Science Service Award.
The annual Wheat Science Service Award recognizes individuals that have contributed to the success of the Wheat Science Group. As a research team, we strive to do the best that we can to address current and future issues facing wheat production. But our best can only be realized with the support of others. We need guidance and new ideas, as well as on-farm research collaborations and advocacy for funding. It is when we work together that great things happen. This year's recipient is Carl Schwenke of Seymour Milling. Carl is a longtime advocate and supporter of UK wheat research. As the Vice President of Grain Supply for Seymour Milling, Carl has been an essential line of communication that addresses issues related to wheat production and end uses. Carl's ideas have steered the direction of UK wheat research. One researcher said, "Carl is one of our biggest supporters. You know when you talk with him that you will walk away with a new challenge to figure out. They are generally complex and in some cases career challenges that require significant work but always result in what's best for Kentucky's wheat industry." He keeps all of us in the Wheat Science Group focused on what's best for Kentucky's wheat industry, starting with farmers and ending with consumers. I truly think his support and challenges have been one very large piece of the foundation that makes and keeps the Wheat Science Group so successful and nationally recognized. Another researcher said, "Carl understands the wheat supply and value chains better than anyone I know. His knowledge, coupled with his friendly demeanor and his understanding of production agriculture, has made him a great friend to the entire wheat industry in Kentucky. He has been a diplomatic bridge between growers and end users, and has helped growers and researchers alike to understand that we are all on Team Wheat. And for the crop to survive, we need to pull together." Carl is also an ardent supporter of wheat research and extension work that is carried out by the Wheat Science Group and has been a great asset to us. And with that, it is an honor to present Carl with the 2021 UK Wheat Science Service Award. Congratulations, Carl.
All right, I just want to second that. Congratulations, Carl Schwenke. And I want to add to some of those comments that Bill just had. I actually first met Carl about 14 years ago when I started at the University of Illinois. Many of you may know, I was extension plant pathologist at the U of I before coming here to UK in 2015. And Carl was one of the very first people that called me. And I think, Carl, you may remember this, but I think you said, I hope you spend some time on wheat. I remember you saying that, and I have, and so I've always appreciated your comments, Carl, and was happy to know that I could continue to work with you when I transitioned over to the University of Kentucky. So with that, Carl, I'm going to hand the mic over to you and let you say a few comments if you'd like.
Thank you, Carl. Can you hear me now? I don't know if you can hear me. Okay. Well, I thank you very much for the award. I want to just comment on a couple of things that you all have to understand that the UK wheat program is a nationally recognized program, and a lot of people have had a lot of work on doing that. But I think some of the key things that have helped that happen is that the interaction between producers and the extension group. There's an expectation that the extension group helps the farmer, and the farmer expects that out of it. And there's been a lot of people that don't want to let that have worked very hard to make sure they don't let the producers down. I mean, there's an awful lot of people that have gone before me, Don Holcomb to name one, Curtis Hancock, Laura Noth, Trevor Gilkey, the previous winners of this, and it's such an honor to be included in that group. But I do want everyone to know that the reputation of Kentucky and Kentucky wheat producers and Kentucky wheat intelligence is high on the list. It's what we buy wheat from 4 different land grant areas, and by far Kentucky stands head and shoulders above any one of those on the wheat and the effort. The other comment I'd make is that we've had a lot of good people involved in the past, and the list is endless. Lloyd Murdoch, Dave Van Sanford, Doug Johnson, Don Hirschman, some of those old guys. But the future for the great thing is the future of the young people that are coming on and involved in agriculture. And I thank Dr. Cox for that. She does an awful lot of it's hard for her. She's as a land grant college, she's got to keep agriculture in the forefront, and that's a hard job to do. And Kentucky has done a great job with that. And I encourage the producers to keep leading leading that charge, because without the producer pushing this, it'll be increasingly difficult for the University of Kentucky to continue their mission that they have. Again, thank you very much, and thank you for all the producers that have done such a great job in Kentucky.
Thanks, Carl. Again, congratulations and well deserved. So we're going to, and yeah, everybody can do the virtual clapping if they would like. Let's see. There we go. So let's transition now over into the panel, and I can see the chat box now. It looks like there's a lot of things in there, so I need to open that up. I'm just going to ask all of the speakers that presented today if they could turn on their camera. And so just as a reminder, we have myself, Dr. Carrie Knott, Bill Bruning, Raul Villanueva, Travis Legleiter, Amber Herman, and Sam McNeil are all available for questions. And so I'm sorry, I'm just getting to see the chat for the first time here, so I'm kind of scrolling here real quick. So it looks like there was a question. I have seen instances where the same variety has different tolerance reactions to Metribuzin. What are some possible reasons for this? Bill, since you had presented on that, do you want to try to tackle that question?
Yeah, I think that's a great question. And, you know, theoretically, if you have a given field with a given variety and a given rate applied, you know, at that precise stage, there should be no differences. But I think a lot of times what we face is, and certainly seed companies probably run into this question, is if you have one grower on, you know, one side of the street that has variety X, and it looks great, and it's been treated with Metribuzin, and you see the person across the street, another one of your clients, is seeing damage from Metribuzin in the same variety right next door. You say, well, what causes this? And I think there's a number of things that can cause this. Certainly the rate of application. You would hope that you'd be comparing apples to apples there. The timing of the application certainly can be related to, damage can be related to the developmental stage of weed at which it's applied. So even if you're applying, say, 4 ounces per acre on both fields, if one field was planted a few weeks later, there's been reports of later planted wheat being more susceptible to injury. So that given variety, which might be considered tolerant, if it's been under some sort of stress, possibly due to late planting, or even winter cold stress damage or ice damage, which can weaken the wheat crop, can leave that susceptible to the Metribuzin or really any chemical damage as well. So I think there's a number of different things. Certainly in the 2 years of data that I conducted here, most of the varieties that were more susceptible seemed to be more susceptible in both experiments. The ones that were more tolerant, but there were a couple outliers that stood out that may have just been due to experimental error, but there were a few that just popped out that looked to be in 1 year moderately susceptible, and in the second year moderately tolerant. So I think there can be some instances there. In different years, I know University of Tennessee originally applied 6 ounces per acre, and they found minimal damage to wheat varieties, but then when they repeated it, they did a 6 and a 12 ounce application, and the following year that they did that, the 6 ounce caused severe damage in many different varieties. So in 1 year, you had a situation where the varieties seemed like they were pretty tolerant, and the following year, there was damage. So I think it highlights the importance of doing multiple year trials and over multiple locations to really look at the effect, environmental effects on how these varieties tolerate really any chemical in general. But really, I think it probably has to do more with just slight differences in the rate of application or the timing of application related to the developmental stage.
Okay, thanks, Bill. Travis, as the weed scientist, do you have anything to add to that?
Yeah, I think I agree with Bill. It kind of, and I think one thing you have to think about is not only the developmental stage, but also what is the environment. So Metribuzin, like many of our herbicides, or almost all of our herbicides, are metabolized by the plant. Typically, the reason a plant is there's a couple reasons why a plant can be tolerant to a herbicide. Sometimes it's because the pathway, the herbicide just doesn't fit the pathway, but a lot of times, with most of our crops, the reason they can tolerate a herbicide at certain rates is because they metabolize it. So, and a plant can't metabolize if a plant is weakened or if the environmental conditions are not ideal for that plant to be growing quick or be growing well. It doesn't metabolize that herbicide as quickly. So I think in a lot of these cases, that's usually what you're dealing with is just environmental conditions that are not ideal for that plant to be able to metabolize that herbicide quick enough, and then you end up getting some injury from it. So I think a lot of times that's probably what it is environmental conditions. And as Bill mentioned, a lot of that's also the stage of the crop as well. So a smaller crop doesn't metabolize as fast as a bigger crop. So.
Okay, thanks, guys. Looks like there was a question for me that came on is, do I have yield data I can share for the fungicide trials? Yes, I do. So I think, well, first off, if anybody has any questions about the fungicide data, they're welcome just to email me directly and I can provide you with some information. carl.bradley@uky.edu is my email address. But I think those are also in the UK Wheat Science Research Reports that are available online at the UK Wheat Science website. So if you just did a Google for University of Kentucky Wheat Science, that should take you to that page, and then you can find the most recent UK Wheat Science Research Reports, and I should have yield data in there. I'm pretty sure that I do. Let me just scroll on down here. So Raul, looks like there was a question from Dave Van Sanford, our wheat breeder. He says in our breeding line grain samples, we use diatomaceous earth for weevil control. We are not in a position to use phostoxin and similar compounds. Do you have any experience with diatomaceous earth?
No, Dave, I haven't used diatomaceous earth for stored grains. However, I work those with plants, and they have some control of immature forms of insects. I believe that some magnesiums have some information about that, especially so we were talking about diatomaceous earth, and it says that this diatomaceous earth can cause some damage to the meals. I don't know if Sam is somewhere over there.
Yeah.
Okay.
Good morning. In keeping with Carl's keeping with Carl's comment. Now so when on Raul's slide he mentioned insecto, that's diatomaceous earth.
Mm-hmm.
Yeah. So I've heard that it's you got to be careful and check with your buyer as a farmer to check that product before you apply it, because they might create a problem down the road or to the processors. Diatomaceous earth is very abrasive and can damage milling equipment. So for that reason, it's not desired in the product itself. You can use it on the bins and certainly under the floors, and that's probably the best application for diatomaceous earth. It needs to be kept dry to be effective. So keep those things in mind. But yeah, I mean, it's a good product. It does control insect activity quite well, but you just need to be careful with where you do apply it.
Okay, thanks, Raul and Sam. Raul, did you have more to add?
Yeah, this is very specific actually for immature forms of insects, because this is a crystal. Actually, it is abrasive, so it kills the insects, making ruptures to the skin. So.
Okay, thank you. Again, if you have questions, feel free to go ahead and put those into the group chat. Sam, since you're on here, I might ask you a question from Raul's presentation. Just wondering, is there ways to monitor? Is there any kind of equipment or anything that can be used to monitor insect activity without having to climb into that bin?
Yeah, there are some tools that you can use. In the past, I've used a pitfall traps, which you would they have to be inserted in the grain itself. But now to do it remotely, there is an instrument that can be inserted in the grain initially, and then you can monitor, because it has the traps themselves has an electronic counter at the base of the trap, and it will count insects as they fall into the trap. Then you can set that up to monitor the activity remotely. Another indirect way to measure insect activity or mold activity, if you have concern about Fusarium activity in the bin for wheat, I think you may want to consider a CO2 monitor, because if insects or molds are active in the stored grain or stored wheat, they'll produce CO2, and there's been, we've been looking at that for a while in the last 10 years or so in research, but it hasn't really gained a lot of attention, seems like, until recently, that CO2 monitoring is more of I think it's going to become more in vogue to use those monitors. And so what you're looking at there is any levels below 400 parts per million would not be a concern, but when it starts creeping up around 1500 or higher, you want to certainly start paying attention. And if it gets much above 3000, then you probably need to turn the grain, or maybe you want to do it before then to control insect activity.
Just I want to add, so you can use also pheromones, especially for weevils and sometimes for the moths, and sticky traps. So there are some issues that you need to monitor your insects on beans, and those can be done inside and outside the beans.
Okay, thanks, guys. I had a private chat come in to me about economic benefit of fungicides, and if that's something that we've looked at yet, and we need to put some numbers to that, and so I don't have that calculated yet, but that's certainly something that we plan to do when we get the chance. I like to work closely with Jordan Shockley, one of our ag economists, and we'll try to put some numbers to some of that and get a better idea about economic benefit with some of the fungicides. Again, keep putting some questions into that chat box if you have any. Carrie, I know there was probably some late planted wheat this year, and as there usually is because of weather. Is there anything that if somebody ended up planting late, is there anything they can do now to kind of help go ahead and still get the best yields that they can get?
Right. Yeah. So like I said earlier in my pre-recorded presentation, really our option now is just mitigating for thin stands, and that would be looking at nitrogen management. So if we have the thin stands, which really are, when I think of thin stands, I think of less than 70 stems per square foot prior to Feeks 3. So some people might already be out. I know my crew started, we'll start next week looking at stem counts as well. So it's not too early. It's a little early, but it's not too early to start looking at those stands and trying to figure out what you're going to do with nitrogen. And so I would recommend maybe a 50-50 split between Feeks 3 and Feeks 5 to help mitigate. We do have some ongoing research. This coming year will be our second year of it, looking at some additional options to mitigate thin stands and late planting. And so I want y'all to stay tuned, but we don't have a whole lot after just 1 year just yet.
Okay, thanks, Carrie. So Travis, are there any alternative herbicides that can be used for control of ryegrass that is resistant to axial?
Not really, unfortunately. So At this point in time, I mean, we consider most of our ryegrass to be ALS resistant. So actually right now, another part of Amber's project is she's done one run in the greenhouse and she's getting ready to start her second run here this semester. But we're screening 22 samples from across the state of Kentucky for herbicide resistance, seeing how much ALS resistance and pinoxidin resistance we have. But if you don't have axial or pinoxidin, really, you don't have much. You can try something like Finesse or PowerFlex, but you probably are not going to have very good luck with it because we do consider most of our ryegrass to be ALS resistant. So you can try it, but I guess I don't have a lot of faith that those are really going to do much for us because they are ALS inhibitors. So that's a reason why we're focusing so hard on the weed seed control, is because it's kind of scary to think that if you don't have that axial product available to you, you really don't have any post-emergence options. And so it's trying to find an alternative to that.
So herbicide resistance continues to be an issue for pretty much all crops that you work on, I think, Travis, right?
Yep. Yes.
I'll segue real quick and just make a comment. Fungicide resistance is also something that we're concerned about. And in soybean, we actually have an issue, frog eye leaf spot, that is resistant to strobilurin fungicides. We've started to look in wheat now, thanks to some funding from the Kentucky Soybean Growers, Small Grain Growers Association. And we've been looking at the one of the leaf blotch pathogens, the Staganospora nodorum leaf blotch pathogen, really common. It also causes gloom blotch here in Kentucky as well on wheat. And unfortunately, I have some bad news. We are finding strains of that fungus that are resistant to strobilurin fungicides here in Kentucky. And so that's one of the things that we're concerned about. And if you notice, I guess I didn't have enough time to go into my presentation a lot about the different active ingredients that are present in some of these scab fungicides. But for a number of years, we've been relying a lot on triazole fungicides. So first starting off with Folicure, and then Prosaro, which contains a couple of different triazole fungicides, and then Carumba, which is a single triazole fungicide. Now the new product, Metribuzin Ace, it also uses a triazole, but it also brings in a different chemistry class known as an SDHI. So this is the first time that we have a scab fungicide that's using something in addition to a triazole fungicide. So there is concern about the scab fungus developing resistance to triazoles. A few years ago in New York, there was an isolate of the Fusarium fungus that was found to be resistant or less sensitive to tebuconazole, which is the product that's in Folicure and one of the actives in Prosaro. And actually, one of our colleagues here at the University of Kentucky, Dr. Kirsten Wise, who came from Purdue University, came here in 2017, but was extension plant pathologist at Purdue. Before that, she just recently published a paper where a few of us from some other states are co-authors. And when I was at the University of Illinois, I sent Kirsten some isolates of the Fusarium fungus that causes scab to her to evaluate. And as it turns out, one of those isolates was resistant or less sensitive to the active ingredient in Folicure, as well as the active ingredient in Carumba. So that was the only one isolate out of several that she evaluated. So pretty low percentage out there. But that is something that we certainly need to be concerned about, because we really have been using triazoles a lot for head scab. So it's good to see a product that has a different active ingredient. I know that companies are also working on some additional experimental products that will be looking at some different active ingredients.
So Carl, can I ask some questions about what you just said? Sorry to jump in.
Absolutely. No, that's great.
So the isolate in Illinois, was it in like a major wheat producing region, or was it just kind of a weird, strange strain, you think?
So it was in Southern Illinois. It was from, I believe, before mentioning this, it's always a good idea to read the paper again before you mention stuff. I believe it was in Fayette County, Illinois, which would be Brownstown, Effingham area. And that was at the Brownstown Research Center that is now closed, but U of I used to have a research center there. That's where that came from. It came from one of my research trials that I had there. And so that's kind of where we were collecting a lot of the heads when we sent to Kirsten, as well as some commercial fields. But that would have been, a field that about every other year we would have had a scab trial there in that particular field. So yeah, it was in a wheat production area. And so that isolate was collected, I think, in 2012. So we really need to kind of get back to monitoring because what really increases the population of resistant isolates is continued selection pressure, which means continued applications of triazole fungicides. So
do we have any surveys in Kentucky that you know of?
We don't. So Dr. Nathan Klacheski, who was at the University of Illinois and who's going to be leaving here at the beginning of February, had a project that was going to be looking at SDHI fungicides, like the one that's in Metribuzin ACE. But there hasn't really been any ongoing work looking at triazole sensitivity in this area. We may pick that back up, especially trying to characterize that one resistant isolate just to figure out what mutations were present in it. But yeah, and again, so far everything looks like it's working, out in the field, but keep in mind, that risk is out there, and that is one of the primary ways that we manage Fusarium head blight. Some really good comments that I was looking through in the chat box, not necessarily questions, but just good comments. And one of those was kind of pointing out the effective line of defense for scab is resistance, just kind of like what I showed, resistant varieties, as I showed in some of the data that I presented today. And so again, that makes it even more important that we're not relying solely on a fungicide to manage that disease, that we really need to be using our most resistant varieties that we have available to us. And one of the things that I left out of my presentation, mostly due to time, it's hard for me to give a 15-minute presentation, I must admit. I had to delete a lot of slides for that. Is where in the heck can I find out about how resistant or susceptible some of the varieties I've been planting is? And of course, companies often provide ratings on that. But one of the really good ways that you can find out and kind of compare varieties is looking at Dave Van Sanford every year has a misted scab nursery, as we call it, and he evaluates all the varieties that are in the UK variety testing program, and those show up in Bill's reports online, as well as in the hard copy report. And Bill also does some evaluations. If scab shows up in a location, he'll do those ratings. So that's one of the places. The University of Illinois does something very similar, that we share a lot of common varieties between Illinois and Kentucky. So there's a couple of good places that you can go to kind of look at how susceptible some varieties might be. And again, that's really the first step in managing this really important disease. As you can see from that data that I presented today, that you got to start, if you start off with a really sound, moderately resistant variety, and then apply a fungicide as well, then you really can drop some of those DON levels down pretty low, which is really what we need to do. So again, if you have questions, go ahead and put those in.
Can I ask another question, Carl, for Raul? Yeah. So Raul, the last few years, we've been looking at different planting dates in my program, and kind of it's based upon the planting dates I'm seeing and hearing from producers. And so a lot of people are moving planting dates very early. And when I say very early, I'm saying, for our region or the southern tier, early October. Traditionally, we've always said, do not plant prior to the Hessian fly-free date. I mean, I kind of want your input on that. We haven't, I'm going to knock on wood, we haven't really had infestations in many years here in Kentucky. And so what are your thoughts on pushing these planting dates in terms of Hessian fly-free? Because the producers are capturing some really nice yield benefits from those earlier planting dates. You're muted.
I'm here since 2016, and I haven't seen any damages due to that pest. So I mean, so I cannot even try to do any test with that one. So, but we need to be precautious. I mean, so if you need to do that early sampling, early planting. You not only need to look into the Haitian fly, but it means you may have also some problems with aphids, because during that period of time we are going to have a very rapid growth population of aphids, and viruses can be an impact. So in addition to that, probably we have some cultivars that are resistant to Haitian flies. So I mean, that's something that farmers should know. And I'm not so sure if Dave can have something to say about this. So, or even you work with different varieties. Do you have any cultivars that do you believe are resistant or tolerant to Haitian flies?
Not that I'm aware of. I don't know about Dr. Van Sanford, whether they really screen for it, right? Because it's so sporadic. You know, from what I'm hearing from producers and other researchers, some of the old timers, so to speak, like Lloyd, you know, we really haven't had an infestation in over 30 years. So I'm just curious. Dave, do you want to pop in on this?
Yeah, I mean, basically, the serious Hessian fly activity occurs to the south of us. And even though for many, many years, Purdue was sort of an epicenter of Hessian fly research, there's very little that is seen north of the Mason-Dixon line. So Georgia, the Carolinas, big problems. But we've had just extremely sporadic occurrences in Kentucky. I think the last one that I was involved in was probably 7 or 8 years ago, when I was asked by Steve Hunt to come take a peek at a field where something weird was going on, and that's what it turned out to be. And there was nothing really unique about it in terms of super early planting date or anything. So there's no particular pattern to it. But yeah, you're right, Carrie. You summarized it well.
Yeah, I think in that 1 year, like I said, I've only seen it that 1 year since I've been in wheat for 15 years. And I did try to take some ratings. And for what I was seeing, I just had a really hard time making heads or tails out of really what I was seeing, and could I really see a difference really anywhere? And I couldn't. So I think it's tricky, unless you're used to seeing it every year and you really get a good feel for it, and really sit down and really investigate these differences in varieties. But for me, I couldn't do anything with it when we did see some of it pop up 1 year.
Carl, could I make a comment?
Yeah. Oh.
Just kind of a perspective of somebody that we buy wheat from all over the region, and Kentucky is, the wheat management has gone so far in the last 20 years. But for much of the early wheat planting management strategies, the only strategy anybody had or tool they had was the fly-free date. So that was the only thing we used, farmers used. That was the only, it was the only thing available to say what you should do or shouldn't do. Otherwise, it would just plant it, harvest it, it was fine. But the Hessian fly, of course, is the average frost-free date, or average freeze date, I believe it is. But nothing replaces management tools like good scouting and looking at it to see if you've got a problem. I would add that this last year, early planted wheat caused Kentucky growers an awful lot of money. We lost 3 million bushels of wheat because of the freeze damage that came through. And much of that, I believe, and you talk to the agronomists that are out there that actually looked at the fields and managed those, I think they would say that most of those problems came from early varieties being planted early. And so there's a lot of other considerations that go on with the fly-free date or the planting date or anything like that.
So just an additional comment, and you already mentioned that, Carl. An extra effort should be done to scout when you do an early planting. So for aphids, even for diseases probably, but it means aphids and Haitian fly, and even probably some wild worms. Yeah.
Yeah, that's a good comment, Raul. And from a disease control perspective, that fly-free planting date is also important. As you mentioned, aphids that could potentially infect wheat with viruses, but also we see powdery mildew is more of a problem if you are planting before that fly-free date. So I think it's still a good thing to observe, and for a lot of reasons, maybe not necessarily the Hessian fly, but I think that's something that growers probably should still consider, that fly-free date, in my perspective, at least. Any more questions? I don't see anything else on the chat box right now. Raul, so we were talking about Hessian fly, and of course you talked about insects in stored grain. You know, why is it important to try to identify what insects are? I mean, if you see a bug, it's a problem, right? Do we need to know what it is? I say that facetiously.
That's correct. So we need to identify the insect properly. So because the beetles, especially the ones that you have in storage grains, are hard to identify with the naked eye. So in some cases, you need to use a hand lens, or even you can be able to send those to your county extension agent, and that county extension agent is going to reach it to me. Also, you can do some images, taking some pictures with your camera. So if you do the proper identification, you are going to be able to have the effective insecticide to control this pest, but also sometimes the rates are different. So that's very important. And just for the, that also, if you identify the insects, you can discern how good is the quality of the grain, because you have primary and secondary feeders. So usually those secondary feeders are feeding on grains that are broken or something like that, whereas the primary feeders, like the ones I show you, they feed in grains that are whole grains, and they can make damage to the grains stored. So.
Okay, thanks. Going back to that freeze event, a couple of freeze events that we had last spring, I did have some questions about any effect that we saw on diseases because of the freeze. And Carrie and her crew were nice enough to go out and actually rate some of my fungicide trials for freeze injury. And that let us kind of take a look to see if there was any kind of correlation with some of the scab damage that we had. And in fact, we didn't see any kind of statistical significance there with freeze injury and scab injury, head scab, Fusarium head blight injury. Although one of the interesting things that we did find on our farm here in Princeton, and in some of my plots, as well as just some of the production wheat on the farm, was ergot, which many of you may be familiar with. Ergot is typically considered to be more of a problem on rye because rye is outcrosses, and those ovules stay open longer on the rye head because it has to take pollen from a different plant to fertilize it. And once the ovules fertilize, it's no longer susceptible to the ergot pathogen. So what happens when you have freeze damage? If those heads become sterile, then that really allows that ergot pathogen to get in and cause infection. And so the reason ergot is a real problem is because it produces these things called ergot alkaloids, which is basically, you can kind of go back in history and see the problems of ergot alkaloids, even going back to some of the Salem witch trials many years ago. So that's something that we would want to avoid for human and any animal consumption. And so anytime we have ergot in our head, that's a real issue. But I didn't hear of widespread problems other than kind of just a few areas. And so I don't think that was a big problem. Dave Van Sanford, I see, commented that they did see a fair amount of ergot in Lexington in both wheat and rye. So there probably was a little bit out there this year because of that freeze damage. There's really not anything you can do about it. Fungicides aren't really effective. And so we just have to try to do things to hopefully avoid those weird freezes. And I don't know if you were on Tuesday or not, but gosh, I just lost track of his name. Our weather person that presented. Forgive me.
Matt Dixon.
Matt Dixon. Yeah.
Thank you. He presented some information. Yeah, I think Matt Dixon. There we go. Sorry, Matt, if you're on. But he presented some information showing that was really a rare occurrence to get that late of or the timing of the freezes that we got this last year. And so hopefully that'll be another 50 years or so before that happens. We have just a few minutes left. And so I think what I want to do here is just kind of toss it around to all of our speakers today to see if they have any final points that they want to make. And so Travis or Amber, is there anything that you would like to go ahead and throw in?
Yeah, I'll throw something in here. So one thing in the video, I showed the two different plots, Anthem Flex and Fierce. And really what I was trying to compare there was really the timing on those. So just to be fair to both of those, because the reality is we had some other plots with the Anthem Flex that we put on 14 days pre-plant. It's not labeled, but we were just looking at it as an experimental, and it also didn't look good when it didn't have an activating rainfall. So I just want to reiterate that point, that it's really more about the activating rainfall. So that's the key to these residuals, any residual in any crop. It's all about the activating rainfall. So I know you guys can't make it rain, but just pay attention to that when we're doing that, or it might help us understand why we might have some failures with some of these pre-emerge herbicides. And I shouldn't say failures, but just not as good a control as what you might have expected. So, but there are several of those out there. So just wanted to reiterate that.
Thanks, Travis. Carrie, do you have anything that you want to share?
I don't think so. I'm just really optimistic for the year, as always. This time of year, I'm always loving the way the wheat looks and ready for some good things to happen.
I think we all are. How about Raul? Do you have anything else? Any final comments?
No, the only thing is that working in store grain is a completely different issue compared to working in field insects. So my experience in this type of deal is a little bit short. So I hope to start doing more work into this starting now. Actually, we just have some hard time to hire people to help in conducting this study. So.
Okay, I'm going to go ahead and I'm going to bring up, I was reminded that I really needed to do this, bring up the part of the reason why many of you are probably on here today. CCA credits and pesticide credits. So if you are wanting the pesticide applicator hours, hopefully you can see my screen right now. You can either put it in the Zoom chat. We need your full name and applicator number, or you can email Colette, and you can see her email address there on the screen with your full name and applicator number. And then if you are a CCA looking for those continuing education units, you got the QR code there on your screen. Now, I'll tell you that only works if you have the CCA app on your phone. If you have the CCA app, then you can go ahead and scan that QR code, and that'll allow you to instantly get credits for this meeting today. If you don't have the app, in the Zoom chat, you can put your full name and number, or just email Colette with that information, and she'll put you on the list, and that'll get sent in to the CCA group. Let's see. We're just pretty much at the end, but I may just go ahead and call on Bill and see if you have any final comments, Bill.
I guess just really in terms of the rate of Metribuzin applied, I mentioned I applied the maximum rate of 10 2/3 ounces per acre. The recommended rate in the fall at the 2-leaf to 2-liter stage is actually just only 2 to 3 ounces. So most growers aren't applying that type of heavy rate. So again, I was applying the maximum rate, which would actually be for wheat that was over 4 tillers. So I think that's something to consider. We have done some preliminary screening of Metribuzin tolerance, Jim Martin and I, a few years ago, where we applied 3 ounces per acre, and we only saw almost all the varieties show no sign of injury at all, with just a few showing some moderate or minimal injury. We also did an 8-ounce rate, where that same year we saw a substantial injury and moderate injury with just a few that weren't injured. So I guess my point is that the application rate does have an effect on how much damage occurs among these varieties.
Okay, thanks, Bill. And I think I'll make one final comment as well. I talked a lot about disease, but that is near and dear to my heart. So I'll make one more comment. So hopefully you got from my presentation that growing moderately resistant varieties is sort of the #1 thing you can do to manage scab. And oftentimes I get a question about, do I have to give up yield or any other important traits to get scab resistance? And the answer seems to be no. I think you can look at some of the data from Bill's UK variety trials, as well as Dave Van Sanford's scab nursery, and then you can kind of pick some of those varieties that look really good for scab resistance, and then kind of see how they performed in regards to yield and other traits. And you're not giving anything up really whenever you're choosing some of those moderately resistant varieties. You can choose varieties that have moderate resistance to scab, as well as bringing in a lot of yield. So that's certainly a way to go. With that, we're just a couple of minutes past what we said we'd be. So we're going to close. And I really want to thank you all for attending. And I think we all are looking forward to attending in person when the opportunity arises, hopefully sooner than later. And I also just want to thank a couple of people behind the scenes that really helped out, including Colette and Chad Lee, who were kind of in the background helping me out and doing a lot of things that you may not have been aware of, but appreciate all that. So again, thank you so much, and looking forward to a great 2021 growing season. Thank you.